by Zaamena Manji,
Kelsey, McCurdy,
and Ignacio, Madrid.
For our investigative project, we performed 2 experiments: the effect of diacetyl on MX52 and wild-type strains in C. elegans through chemotaxis assay, and examine location of sensory neurons in MX52 and wild-type strains of C. elegans through neuron morphology dye-filling assay. And finally, the observation of MX10 strain of C. elegans using a fluorescent microscope. C. elegans are free-living roundworms about 1 millimeter in length. They are used as a model organism in genetics because they are easy to maintain and have a short life cycle and produce large number of progeny. C. elegans was the first multicellular organism to have its whole genome sequenced. The study of C. elegans is relevant to humans because approximately 70% of human genes are also found in C. elegans. C. elegans genes can be mutated through a process called mutagenesis, which is the use of chemical reagents or radiation to alter the gene's DNA, and then mapping the sequence to determine the mutation. For our investigative project, our goal was to characterize the BBS8 gene in C. elegans. The human ortholog, BBS8, is mutated in an autosomal recessive and genetically heterogeneous disease called Bardet-Biedl syndrome. This disease causes retinal degeneration, obesity, malformations, polydactyl, and learning disabilities. BBS8 encodes a protein that is part of a bigger protein complex called the BBSome, which is involved in trafficking signaling receptor proteins to cilia, which is a microscopic organelle that extends from the surface of cells. First, to determine where BBS8 gene is expressed, we used a fluorescent microscope to image an individual MX10 strain C. elegans. This strain contains GFP gene, or green fluorescent protein gene, that serves as a reporter gene to assay for BBS8 gene expression. Reporter genes can be directly attached to a gene of interest to create gene fusion. This is useful when your gene of interest has little or no obvious observable effect on the organism. The 2 genes are under the same promoter elements and are transcribed into a single messenger RNA molecule. The mRNA is then translated into protein, where both the gene of interest and the reporter gene are expressed. In our project, we characterized the C. elegans strain MX52 that contains an 819 base pair deletion in the BBS8 gene and compared it to wild type. In C. elegans, BBS8 is located on chromosome 5 and encodes for a tetratricopeptide repeat, also known as TPR. TPR is a protein-to-protein interaction module found in many functionally different proteins that facilitates a specific interaction with partner proteins, as well as mediates the assembly of multi-protein complexes. In C. elegans, previous research has shown that BBS8 activity is essential for cilia biogenesis and function. Therefore, a mutation within BBS8 leads to odorant chemotaxis defects as well as abnormal motility. BBS8 is also necessary for proper regulation of insulin secretion.
We conducted a chemotaxis assay to test the difference in attraction to diacetyl between wild-type and BBS8 mutant C. elegans. Chemotaxis is locomotion towards or away from chemical stimuli. In preparation for the chemotaxis assay, wild-type and mutant worm plates were washed in 1 milliliter of S-basal buffer and then collected and combined into a 15 milliliter conical vessel. After collection, the worms were centrifuged at 2,000 revolutions per minute for 2 minutes in order to form a pellet at the bottom of the tube. Then, excess M9 buffer was removed to a final volume of approximately 200 microliters. After the removal of the buffer, the worms were then washed again in 1 milliliter of M9 buffer, centrifuged, and excess M9 was removed. The repeated washing was to ensure the removal of any remaining bacteria. The tube was then resuspended in order to obtain a homogeneous mixture. 6 chemotaxis plates were prepared with approximately 25 to 100 worms pipetted into the origin of each plate. Immediately after, 2 microliters of the test solution, which contained 0.5% diacetyl, 25 millimolar sodium azide, and ethanol, and the control solution were placed into their appropriate quadrants. The plates were left for 1 hour to allow chemotaxis, and after that hour, the plates were examined and the amount of worms was quantified to calculate the chemotaxis index. In order to examine the structure and the function of the sensory neurons, as well as the ability of the C. elegans to uptake lipophilic dye in the neurons, a neuron morphology dye-filling assay was performed. The purpose of this assay is to determine if the development of sensory neurons are normal. Performing the assay can also demonstrate the proteins and genes that are associated with the function and development of sensory neurons. 2 plates containing wild-type and mutant worms were obtained. 2 milliliters of M9 buffer was pipetted onto each plate, swirled and tilted to remove the worms from the agar. The worms were gathered into a 15 milliliter conical vessel. The tubes were then centrifuged at 2,000 revolutions per minute for 2 minutes in order to obtain a pellet of worms at the bottom of the tubes. After being centrifuged, the excess liquid was removed until approximately 500 microliters of the worm buffer and the worm pellet were left. 1 microliter of the red lipophilic fluorescent dye, DIL, was added to the tubes. The tubes are then covered with aluminum foil in order to protect the fluorescent DIL dye from reacting with light. The tubes were incubated for 1 hour at room temperature and inverted every 10 minutes for that hour. Then the tubes were spun for 2 minutes again at 2,000 revolutions per minute. Excess M9 was removed until 0.1 milliliters was left in the tube. The worms were washed 3 times with M9 buffer, and after the washes, excess M9 was removed to a volume of approximately 100 microliters. The dye-filled worms were resuspended into the 100 microliter solution, pipetted onto a new plate, and distained for 30 to 60 minutes. After distaining, approximately 20 worms were picked onto an agar pad. The worms were placed under a fluorescent microscope, counted and categorized as neuronal dye-filled and non-dye-filled. The percentage of the worms that were dyed normally and those that were dyed defectively were then calculated, and an image of the fluorescence was taken and data was recorded.
We are now going to discuss the results, conclusion, and discussion of our experiments. In these experiments, the main purpose was to examine the strains MX52 and MX10 and compare them to wild-type C. elegans. The MX52 strain contains an 819 base pair deletion in the BBS8 gene. A mutation in this gene affects the production of TPR proteins as well as cilia cells, causing a defect in the organism's olfactory sense. The MX10 strain is unique because it contains the green fluorescent protein gene, which aids in the assay for the BBS8 gene. To examine these strains of C. elegans, 2 experiments were conducted: chemotaxis assay and neuron morphology dye-filling assay. The goal of the chemotaxis experiment was to examine the affinity or aversion to diacetyl by the olfactory receptors in C. elegans. The goal of the neuron morphology dye-filling assay was to examine the structure and function of the sensory neurons, as well as the ability of the C. elegans to uptake lipophilic dye in the neurons. For the chemotaxis results, it was anticipated that there would be a difference in attraction between the wild-type and mutant worms. However, results were inconclusive. It was expected that the wild-type would be more attracted to the diacetyl, and the mutant worms would have less affinity to the diacetyl because of the mutation in the BBS8 gene, which causes a defect in the sensory cilia cells. Our results show that the chemotaxis index values were relatively low for all strains, which suggests little to no attraction to diacetyl by the C. elegans in the test quadrants. Results of the neuron morphology dye-filling assay indicated that in wild-type worms, a majority showed fully developed sensory neurons in either the head or tail region, but not both. However, in the mutant MX52 strain, a majority of the sample showed developed sensory neurons in both the head and tail. Observing the MX10 strain under a fluorescent microscope allowed the visualization of neurons in C. elegans that the BBS8 gene is genetically responsible for developing. The importance of this research was to examine the stimuli behavior response circuit to the chemical stimuli diacetyl in order to understand the signaling of olfactory cells in humans. The study of the BBS8 gene and its potential mutations could aid in the prevention of the Bardet beetle syndrome as well as other various ciliopathies.